Hello. So I'm going to continue on the theme of genetic linkage, which I started in the last lecture. Today I'm going to talk about how you can use linkage to create genetic maps, which basically just tell us something about the orientation of genes along the length of the chromosome and roughly how far apart the genes are. And then I'm also going to talk about physical maps a little bit and the relationship between the genetic map and the physical map. And what I'm showing here on this first figure is the upper part of the figure shows a genetic map. Genetic maps are based on just recombination frequencies between genes. And a physical map, there's multiple kinds of physical maps, as I'll talk about later in the lecture. One type of physical map is a chromosome that you can stain using a particular dye, showing the banding pattern along the length of the chromosome. And we'll talk about the relationship between the genetic map and the physical map later in the lecture. So let's begin with talking about how you create genetic maps. I'm also going to continue with the theme of last time in the sense that we're going to be using the same model organism, Drosophila, to create our genetic map. But I'm going to add an additional gene to the two genes I talked about last time. So today I'm going to add the black gene, which resides on the same chromosome as the two genes we talked about in the last lecture, the vestigial and purple genes. So just define the black gene. If you're homozygous for the recessive allele black, you end up having sort of uh, a black pigmentation to the body. And if you're heterozygous for the recessive allele black or you're homozygous for the wild type allele, flies have more of a tan color to their body. So I know this looks striped, but we're just going to call that tan in our example. So let's say we set up the following cross where we have a fly that's a dihybrid for the purple and black gene. And again, the purple gene affects eye color. And you do a test cross. So again, immediately when I say the word test cross, you should know what that means is that you're doing a cross to a homozygous recessive for both of the traits that you're looking at. So we do test cross and we get the following results. So we should see immediately from the results of this cross that this is not the result we'd expect from independent assortment, because you expect from independent assortment to see all four progeny classes in equal numbers. And in this case, you can see that two of the progeny classes are much more abundant than the other two progeny classes. The other key that indicates that this is not independent assortment is that the two progeny classes that are much more abundant are roughly equal in number, and the two progeny classes that are lower in abundance are also roughly equal in number. We should also be able to tell from the outcome of this cross what the parental type is. And basically, the way you do that is, as we described last time from this cross, you can look at the most abundant categories, and that tells you what the arrangement of alleles were on the parent that gave rise to these offspring. So in this case, if you look at the parental types, one of the parental types has the purple plus allele, the purple dominant phenotype, and the recessive black phenotype. And the other parental type has the recessive purple trait and the dominant black trait. So the parental type in this case was in the trans configuration with the dominant alleles on different homologs. And then I won't belabor the going through the recombinant types, you can see for yourself, but it's worth looking at that closely to make sure you understand how those recombinant types arose. And then, as I talked about last time, if you want to determine the recombination frequency, it's just a matter of calculating the number of recombinant types divided by the total number of progeny that you see in the cross. So for this example, the number of recombinant types is 307 plus 295. And the total number is I'm just tallying it up below. I think you can do the arithmetic here. This comes out at 0.06, you multiply by 100, and that gives you the fraction recombinants, which is, is 6%. This is also the same thing as saying 6 centimorgans or 6 map units. So you see 6% recombination taking place between the purple and the black gene in this example. Okay, so now I'll tell you how we can use this information to construct a genetic map. So we learned from the last lecture that the purple vestigial recombination frequency is 11.5%, and we just worked out now that the purple-black recombination frequency is 6%. And then if we did additional crosses, looking at the recombination frequency between vestigial and black, what we'd learn is that in fact the recombination frequency there is 16.5%. So Alfred Sturtevant's huge mental leap was basically to sort of say, well, maybe this recombination frequency is telling us something about the arrangement of these genes along the length of the chromosome and roughly how far apart they are. And again, the idea is that perhaps percent recombinations is directly proportional to distance. So if the recombination frequency is low, they're very close together. If the recombination frequency is high, then they probably are very distantly spaced with one another. So he used this information to create a genetic map. And basically what he did is he drew a line that represents the second chromosome, which is the chromosome that these genes reside upon. And he placed the purple gene on that map. And then he sort of said, well, the vestigial gene maps 11.5% or 11.5 centimorgans away. So he drew that on the map. And he spaced those by 11.5 centimorgans. And then we know that the black gene is 6 centimorgans away from purple. So he placed that on the map as well, 6 centimorgans away from the purple gene. Okay, so this is a genetic map. And on the next slide, I'll show you examples of additional genetic maps. So here's some additional genetic maps. So the genes we've been talking about are on Drosophila chromosome 2, so they're actually pictured here. This is a bunch of other genes that have been mapped to chromosome 2 in Drosophila. And these are 4 of the chromosomes from peas. These are genetic maps for these 4 chromosomes. And here's a genetic map of the human X chromosome. So there's lots of examples of genetic maps, and you can basically create these in any organism. So now I'm going to return to something I mentioned at the very beginning of the first lecture I gave, where I talked about these traits, the pea plant height trait and the pea plant pod shape trait. And I mentioned to you that they don't assort independently because they reside in the same chromosome, and that they actually reside very close together on the same chromosome. But now I can actually explain the ratio of offspring you got when you crossed a dihybrid. You did a test cross of dihybrid containing these traits, or these alleles of these genes. So it turns out these 2 genes reside 10 centimorgans apart from one another, so you'd expect to see recombinants occurring at about 10% of the time. So if we go back to one of my slides I gave you in the very first lecture, I told you that the correct answer from this cross would be that you should see a 9:1:9 ratio. And the reason you see that ratio is, again, because only 10% of the progeny would be recombinants. So if you look at this ratio, that's also consistent with this idea that 10% recombination is taking place in this cross. Okay, so why are genetic maps useful? I'll tell you one reason today, and I'll tell you another reason in the next lecture. So one of the reasons they're useful is because they allow you to predict the progeny phenotypes and numbers from a cross. And we'll go through an example of that now. I'll show you how you go about doing this. And the genes we're going to use in this example are going to be the vestigial gene, which we've already talked about, and yet another new gene in Drosophila called the lobe gene. And these positions are where the genes reside on the chromosome. These are map positions on the chromosome. But I'm going to make a simplified map in a minute that basically deals with the issue that we're most concerned about in making predictions from a cross. So again, I already told you from last lecture, the vestigial wild type allele is dominant of the vestigial mutant phenotype, which basically is a wing. The homozygous mutant has a wing-shaped defect. The lobe gene, the wild type allele lobe, is dominant over the recessive mutant allele, and the wild type allele has a normal eye, confers a normal eye phenotype, and the recessive allele results in a deformed eye shape. Okay, so let's make our simplified map. So the important point here is that the distance separating the vestigial and lobe genes are 5 centimorgans. And we're going to use this map to predict how many offspring of a given category we'd get from a cross of a dihybrid that carries these heterozygous alleles of each of these 2 genes. The cross we're going to do, one of the parents is the dihybrid, and the parent is actually in the cis configuration where both of the dominant alleles are on the same chromosome or the same homolog. And the other parent is just going to be a standard test cross parent, so it's homozygous for both of the recessive alleles of these genes that we're looking at. And what we're going to do is we're going to do a cross using these parents, and we're going to count 10,000 progeny. And we're going to predict the progeny phenotypes and the numbers of each type of progeny from this cross. Okay, so first of all, we'll talk about the recombinant types. Based on the cross that we carried out, we can predict both of the recombinant types. One recombinant type is going to have the vestigial plus allele and the recessive allele of lobe. and those are going to be the gamete genotypes of one of the recombinant types, and the phenotype of the flies will be that they'll have wild type wings and they'll have a deformed eye. Again, these are the gametes and the phenotypes. And the other recombinant type is going to be just the reciprocal recombinant, which is going to have the recessive allele vestigial and the dominant allele lobe. And then the phenotypes of these flies will be that they'll have this deformed vestigial wing and they'll have wild type shaped eyes. So what we predict, basically, the number of recombinant products in this interval is we'd see 5% of the progeny, 5% of the 10,000 progeny would be recombinant types. So it's very easy to do that calculation. So 5% of the 10,000 progeny would be recombinant, so we'd expect to see 500 recombinants in total. Now, there's two types of recombinants, so 250 of the recombinants would be of one type and 250 would be the other type. So we'd expect to see offspring with these phenotypes roughly in equal proportions, and we'd expect to see about 250 of each of them. So now with this information, it's relatively easy to calculate the parental types. You just basically subtract 500 from 10,000, divide that by 2, and that's how many of each of the parental types you'd expect to get. And the two parental types are shown here, and I think you can infer what the phenotypes of these flies would be based on what I've already told you about the genetic inheritance patterns of these genes. Okay, so now we're going to go to a question here. Okay, so I'll read the question. Brown seed pods B in a plant species is dominant to green, little b, and elongated pods E is dominant over squished, lowercase e. So the first part of the question is going to ask, a fully heterozygous plant has the dominant alleles linked in trans at a map distance of 20 centimorgans. What will be the predominant type of gamete produced by this plant? So this is basically asking, what are the parental types? So you can pause the lecture here if you'd like and just work this out on your own. It might be helpful to have a piece of paper handy so you can write this out. And then when you're ready to see the answer, just continue the lecture. Okay, so the answer in this case is B, big B, little e, and little b, big E. And the reason that's the correct answer is because, again, it said that the dominant alleles are in a trans configuration. Okay, so part B, what frequencies or percentages will the gametes big B, big E, and big B, little e occur in respectively? So again, you can pause the lecture here if you'd like, and then you can answer the question and then restart the lecture when you feel like you have the right answer. Okay, so in this case, the correct answer is D, 10% and 40%. And the reason it's D, I'll diagram this below. Basically, you're going to get primarily parental types out of this cross, and the parental types are going to be big B, little e, and little b, big E. And you're also going to get a lower frequency, 20% of the time. The non-parental types are recombinant types. So that's little b, little e, and big B, big E. But the question is asking about one of the parental types and one of the recombinant types. And so you have to cut these numbers in half because there's two different parental types and two different recombinant types. So the correct answer is 10% and 40%. Okay, so now I have a third question that I'm going to ask from this, and this is a much more challenging question. So if this plant is self-pollinated, what progeny phenotypes will you expect to see and in what frequencies? And to answer this question, you need to use a Punnett square. So, and the cross is written out here below, and the phase of the alleles is shown in the cross below. So from this information, you should be able to answer the question. If you want to try this as a challenging question, and just pause the video and see if you can answer it, that's fine. But on the next slide, I'm going to actually sort of walk you through the process that you need to use to answer this question. So it's up to you whether you want to try this on your own. It's a fairly challenging question. Okay, so the way you do this is you create a Punnett square just as you've done in the past. And basically what you want to do is you want to write all the different gamete types that you'd expect to get out of this cross. So those are all written on here for you. And then what you want to write below the gamete types, you want to actually write the percentage of the time you'd expect to see each of these gamete types, or the probability of seeing them. And so I'm putting all that information on here now. So these just represent the probability at which you'd expect to see each of these types of gametes being produced. And this is just based on the information we talked about in the first part of the question. And then what you have to do is just fill out the Punnett square as you're normally used to, and you just multiply the probabilities to say what what the probability is of seeing any one type of offspring from this cross. So for example, the first square, that would be the genotype of the offspring, and then the probability at which you'd expect to see offspring of that genotype is just simply 0.16. And so on for the next part of the Punnett square, there's the genotype, and that's the probability at which you'd expect to see that progeny of that genotype. And I'll just do the third one and we'll move on. So that's again the genotype, and I think you guys can answer this question because you've done a lot of these in the past. And that's the frequency at which you'd expect to see progeny of that particular genotype. And then you can total up the progeny phenotypes below, because some of the genotypes are going to give rise to the same phenotypes. Okay, so how does the genetic map relate to the physical map? So as I've been talking about up to now, genetic maps are based on recombination frequencies between genes. So for example, the genetic map we created today, this information is placing the genes along a linear order along the chromosome, and the distance we're spacing these genes apart is just based on the frequency of recombination that occurs between the genes. I basically told you sort of its big insight was that recombination is going to be proportional to the distance between the genes. So if the genes are distantly spaced, you're going to see a lot more recombination. If they're close together, you'd see less recombination. And the assumption underlying all of this is this spacing reflects some aspect of the physical map, or it's the spacing that occurs in the DNA, basically. And physical maps are based on DNA sequences, and there's lots of different kinds of physical maps. I'm not going to go through all of them. I'll just mention a couple of them. One type of physical map is a chromosome, and you can see certain chromosomes in certain organisms and in particular tissues. You can sometimes see chromosomes during mitosis. But basically, this is a chromosome from Drosophila that's been stained with a dye, so you see a series of bands. And so basically, this is a physical map that people have used for many years before DNA sequencing was available. And you could orient where genes were along the length of the chromosome based on various techniques, and you would just position them based on how many bands separated the different genes. So that's one type of physical map. The highest resolution physical map is just simply the number of base pairs of DNA between genes. That's the map we're going to be mostly using in the course. So this is the one I want you to be most familiar with. So again, the highest resolution physical map is just simply the number of base pairs of DNA between genes. So how does the genetic map relate to the physical map? So again, the genetic map is based on recombination units. The main unit that's used is the centimorgan, which is the same as a map unit, or the percentage of recombination that takes place in a particular interval. And the main, or the highest resolution unit of the physical map is the number of base pairs of DNA. So one thing we know about the genetic map versus the physical map is that the order of genes is conserved in the genetic map and the physical map. So for example, if the order of genes that's been worked out from the genetic map is as shown here, A, B, C, D, in the physical map, the order would be exactly the same, A, B, C, D. The other thing is the distance separating loci in the genetic and physical maps is proportional. So for example, if there's one centimorgan separating A and B, and one centimorgan separating B and C, the number of base pairs separating A and B would be about the same as the number of base pairs separating B and C. And if you have 10 centimorgans separating C and D, you'd expect about 10 times more base pairs separating C and D than the number of base pairs separating A and B or B and C. So one thing I will say, though, about this proportionality, this is true in pretty much every organism, but the number of base pairs that corresponds to a given recombination unit varies tremendously in different organisms. So for example, in humans, as I'm showing you on the slide, one centimorgan corresponds to about 1 million base pairs. So for every million base pairs, you see about 1% recombination in humans. But in yeast, one centimorgan corresponds to only about 3,500 base pairs. In fruit flies, the organism we've been using in a lot of our experiments here, it's one centimorgan corresponds to roughly about 600,000 base pairs. So the proportionality is conserved in flies, humans, and yeast, but again, the number of base pairs that corresponds to a given recombinational unit varies tremendously in different organisms. So why are the genetic and physical maps proportional? And the reason they're proportional is because recombination frequency is actually relatively uniform across the genome. So this is human chromosome 12, and what I'm going to plot on here is the rate of recombination that takes place across the genome. And what you can see is basically the recombination is 0 near the centromere, but it's actually pretty uniform over the rest of the chromosome. And that's the reason why the genetic and physical maps are proportional. It's because recombination frequency is fairly uniform over the length of the entire chromosome. Now, this is a little bit of an oversimplification of the true situation. I mean, what I'm telling you here is actually really true. It's fairly uniform over the chromosome. But if you look at it at a much finer level, looking at it over a very small sliding window, what you'll find is a picture that looks more like this, which is that you see these peaks and valleys of recombination rate along the length of the chromosome. And the peaks represent sites where recombination rates are extremely high, and the valleys represent sites where recombination rates are very low. If you average recombination over larger chunks of the chromosome, it's pretty uniform. But there are these hot spots of recombination and these places where recombination doesn't happen very often. The reason I mention this now is because I'll come back to this in a subsequent lecture to talk about this in a little bit more detail. But the important point that I want you to have for now in your mind is that recombination frequency is uniform basically over the entire chromosome, with the exception of the centromere. You don't see recombination taking place at the centromere at all. So that's the main picture I want you to have. Okay, so this is a point where I'm going to transition to something that tends to be a little bit counterintuitive. I think actually linkage is one of the most difficult subjects for students to feel comfortable with, and it's partly because there are elements of linkage that are counterintuitive. So, okay, so let's talk a little bit about the proportionality between the genetic and physical maps in a little more detail. Because it turns out this proportionality breaks down for distantly spaced genes. So if we plot out on the y-axis the map distance, which is based on recombination frequency again, versus the distance apart in base pairs of DNA of genes, what you naively or intuitively expect to see is that this should be a linear graph, basically. The further apart genes are, the more recombination events that take place, and you just expect this linear relationship. But in fact, in practice, when you measure recombination frequency between any two genes, what you find is the further apart those genes are, the less it corresponds to this linear expectation, and it actually tends to plateau at about 50 centimorgans, okay? And it turns out, in fact, for any two genes, when you measure the recombination frequency, that recombination frequency never exceeds 50 centimorgans. Okay, so I know this is very counterintuitive. This is not what you expect. So I'm going to spend a little time trying to explain why that's the case. So why 50 centimorgans? Okay, so let's first consider the situation where we're looking at recombination, just a single recombination event, okay? And we're going to consider 100 cells undergoing meiosis. And the range of possibilities in what could happen from a single recombination event is that either the genes are tightly linked, meaning that they're very, very close together, or the genes are so far apart that the recombination takes place basically every time that the cell undergoes meiosis. All right? So let's just consider the situation out of 100 cells undergoing meiosis, if even one cell has a single crossover event. So if one cell has a single crossover event, you'd expect to see 2 recombinant gametes being made out of 400 gametes that are made in total. Because remember, when 100 cells undergo meiosis, each time a cell goes through meiosis, it produces 4 products of that meiotic event, right? That's why the total number of meiotic products in this case would be 400. So a single cell has a crossover that results in 2 recombinants out of 400. And my little diagram above shows you that this would be the example of that. A single crossover event results in 2 recombinant types. The other ones would be parental. And if all the remaining 99 cells does not undergo recombination between the 2 genes we're considering, they would all have the parental arrangement of alleles. Okay, so now let's consider what happens when 10 cells each have a single crossover. If 10 cells, you'd end up with 20 recombinants out of 400. That would be the same as 5% recombination. And if all 100 cells have a single crossover, so every cell has a single crossover, that would result in 200 recombinants out of 400, and it would give rise to a recombination frequency of 50%, basically. So basically, even if every cell, all 100 cells, had a single crossover, the maximum detectable recombination frequency that you would see would be 50%. Okay? All right, well, what happens when you have multiple recombination events? This is where it gets, this is also very interesting. So we're just going to consider all possible double crossover events. Okay, and there's three different types of double crossover events. The first one we'll consider is a double crossover that involves two strands of DNA. So when you have these kinds of situations, what you end up producing is basically the four gametes that I'm showing you to the right. And it turns out, if you look closely at all four of these are parental type gametes. Okay? And now let's consider a double crossover that involves three strands of the DNA. So in this situation, if you look closely at the products, the recombinant products, what you'd see is that in fact two of them are parental and two are non-parental. Two are recombinant types. And the third type of double crossover that you could have is a double crossover that involves all four strands. And when that happens, what you notice is that in fact all four of the products are recombinant types in this case. So given that all three of these double crossover events are equally likely, on average, what you expect to see is, on average, about half of them are going to be parental and half are going to be non-parental. So, and this is what I'm telling you about this is actually also true if you consider, for example, triple recombinants, quadruple recombinants. You'd still end up with, in the end, having an equal number of parental and non-parental types. So the maximum recombination frequency in any interval is never going to exceed 50%. Now I've got this, if you look at the title of the slide, you can see what is the maximum detectable recombination frequency in an interval. So I've got detectable in parentheses. And the reason that I've got detectable in there is that when we do crosses, we're not actually looking at the chromosomes and saying, oh, there's a crossover event that happened here. So if we, for example, the very first example, you can see that there are two recombination events that took place on those chromosomes. But because we're not looking at the chromosomes to count how many recombination events took place, we're actually looking at the outcome in terms of the progeny phenotypes. We don't see these recombination events, so they go undetected. Okay? So if we go back to this slide that I showed you earlier, intuitively what you expect to see is that as the further apart genes move from one another, the higher the recombination frequency would be. You'd expect to see this red graph where it just increases linearly over time, over distance, basically. So that's exactly what you expect to see. But the reason you expect to see that is that's actually basically what you would observe if you could actually count all the recombination events that were taking place between any two genes. The problem is many of these recombination events are invisible to us because we're not looking at the chromosomes to look at the recombination events. We're actually just looking at the progeny phenotypes to infer whether recombination has taken place. And because of that reason, many of the recombination events, we just don't see them. So another example that hopefully makes clear what I'm trying to say is we made this map of the black, purple, and vestigial genes. So one of the things you may or may not have noticed when I was going through this lecture is that I sort of said if you measure the recombination frequency between vestigial and black, what you'd see is 16.5% of the time you'd see recombinant types from that cross. And in fact, if you count the number of map units between black, purple and vestigial, if you add those together, that adds up to 17.5 centimorgans, okay? Not 16.5. So why is it that these two numbers don't agree? Is it because there's just some experimental error that in our measurement of the actual recombination frequency? And the answer to that question is no, it's not because of some experimental error. It's because when we measure the recombination frequency between black and vestigial, we simply miss some of the recombination events that take place. However, some of the recombination events that we miss when we measure the recombination frequency between black and vestigial, we would pick up when we measure the recombination frequency between black and purple and between purple and vestigial. So to remind you, this is what the chromosomes look like, right? So this is an example of two recombination events we would not detect if we were measuring the recombination frequency between black and vestigial, because this double recombination event does not result in any sort of rearrangement of the alleles. All the progeny types that you would see would be parental types. But if we place the purple gene on here, as you can see, if we're measuring recombination between black and purple, we would see that recombination event that takes place between black and purple, and we would also see the recombination event that takes place between purple and vestigial. So basically, these genes that are very close together, you're much more likely to detect all the recombination events if you're working with genes that are actually close together and measuring the recombination events, or you'll certainly see more of the recombination events at the very least. And it's actually for this reason that if we add together the recombination frequency between black and purple and vestigial, the number actually ends up coming out bigger than the number you expect to see if you just measure the recombination between black and vestigial directly. Okay. So why is it that these maps actually exceed 50 centimorgans then? So you can look at these. For example, in Drosophila, there's 107 map units over the entire length of the Drosophila second chromosome, over 150 map units, and some of the genetic maps from pea plant chromosomes, and there's over 180 map units on the human X chromosome. So the reason that there's more is that people actually there can be more than 50 map units is because people measure the map distance between individual genes, and then they just add these up over time over the just length of the chromosome, and that ends up producing a map that has over 50 centimorgans in it. So to try and make that clear, what people do is they actually try and calculate map distances using closely spaced genes, and those are the values they use when they create their maps. And then if you use distantly spaced genes, it would result in less accurate maps because you're going to miss a lot of the recombination events. So for example, the Drosophila second chromosome, the way this map was made, basically what people did is they took a couple of genes and they calculated the frequency of recombination between them. They're very closely spaced. And what they found is that the frequency of recombination in the examples I've got the arrows next to was 1.3%. And then what they did is they moved to another set of genes, and then they've calculated the recombination frequency between those genes, and then they kept on going along this way until they created the entire map, basically. Okay. All right. So interestingly, some of the genes that Mendel actually worked on were on the same chromosome. As you learned in the first half of the course, that from Mendel's work, alleles of genes assort independently from one another. It turns out peas have 7 chromosomes, and some of the genes you guys talked about in the first half of the course actually are on the same chromosome, and yet they still show behavior independent assortment. So for example, the gene for pea color, yellow versus green, is on chromosome 1. Likewise, the gene for flower color, purple versus white, is also on chromosome 1. And these genes pairs were used by Mendel to show that they assort independently from one another. Another pair of genes that Mendel used in his studies, the gene for flower location, axial versus terminal, which is on chromosome 4, and the gene for plant height, tall versus short, which is also on chromosome 4. And again, in Mendel's experiments, these gene pairs assorted independently from one another, basically just the same as if they were on separate chromosomes. And the reason that's the case is because it turns out these genes are very far apart on the same chromosome. So they exceed 50 centimorgans. So anytime a gene pair on a chromosome is more than 50 centimorgans apart on the genetic map, it's just going to show independent assortment behavior. It's going to act as if the genes are on different chromosomes. You're just going to see 50 centimorgans recombination frequency. Because again, if I didn't make this point clear, the maximum recombination frequency you're going to see between any pair of genes is never going to exceed 50 centimorgans. If they're far apart on the same chromosome, they're going to behave just as if they're assorting independently. I also had this word that I put on on one of the slides in my first lecture where I said genes on the same chromosome do not necessarily assort independently. So it turns out that the reason I use the term necessarily is because they can assort independently, behave as if they're assorting independently, even if they're on the same chromosome. Okay. So to summarize what I've told you in today's lecture, genetic maps just provide information about the orientation and distance between loci based on recombination frequency. And physical maps provide information about the orientation and distance between loci based on base pairs of DNA. And as it turns out, the distance separating genes in the genetic map, which is based on recombination frequency, is proportional to the distance separating genes in the physical map, which at the highest resolution is based on base pairs of DNA. And that's true for closely spaced genes. If you start to look at genes that are very distantly separated from one another, and you calculate recombination frequency between those two genes specifically, what you're going to find is it's not going to be proportional for those distantly spaced genes. So again, the proportionality between genetic and physical maps breaks down for distantly spaced genes, and distantly spaced genes begin to sort independently from one another, or they show that behavior. All right, so now I'm going to have another question for you. And the question I'm going to ask is, where does spec map? So this is yet another gene in Drosophila, and I'm just putting up here the map that we've already created together. And so if you look at the recombination frequency between vestigial and spec, what you'd find is it was 41.5%. So now you have a decision to make. Is the spec gene to the right of vestigial or is it to the left? So I'll give you two choices here. Is the spec gene 41.5 centimorgans to the right of the vestigial gene? That's option A. Or is it 41.5 centimorgans to the left of the vestigial gene? And that'll be option B. I'll give you a third option here too, that you can't tell. So you can pause the video if you want to think about this for a minute, or we can just keep on going. The correct answer is you can't tell. There's no way to tell from this cross where to place spec. We know it's on one side or the other, but we don't know where. So how would we resolve this issue? So what would we expect, for example, if we did crosses of spec to one of the other mutations on here? So if it's to the left, we'd expect to see fewer recombinants with both purple and black. If it's to the right, we'd expect to see more recombinants with purple and black. So let's go through and do one of these crosses then. So we're gonna basically look at the recombination frequency between purple and spec, and that'll help us to figure out where to place spec on this genetic map. So this is the set of crosses we did. We take a pure breeding wild type fly, so it's got the wild type alleles of the purple and spec genes, and we're going to cross that to a homozygous recessive for both of these traits. And we're going to create this F1 dihybrid. Based on the cross that was carried out, we should know the phase of the alleles in this dihybrid, and we're going to do a test cross. Again, you know what the test cross genotype is. It's homozygous recessive for both of the alleles both of the gene pairs we're looking at. And so we get the following progeny out of this in the following numbers. So we get 2,500 of the parental types roughly, and then when we look at the recombinant types, we get about 2,500 of each of those as well. And again, you should be able to look at this cross and understand why I'm calling these the parental types and the other ones the recombinant types, okay? You should know the phase of the alleles, and that should tell you which are the parental types and which are the recombinant types. Okay, so you can just look at this cross, and basically this tells us that the purple and spec genes are essentially behaving as if they're on separate chromosomes. This is also telling us that the reason it's telling us that is because the number of parental types and the number of recombinant types are basically the same. So this tells us that these genes are at least 50 centimorgans apart on the genetic map. So now we can go back to this map again, and I can ask you, which side of the purple gene should we place spec? Should it be to the right, option B, or to the left, option A? And the correct answer is option B, because remember, the number of map units separating vestigial, if we add up the number of map units separating vestigial and spec and purple and vestigial, it adds up to more than 50 centimorgans. So we place it to the right. If it was to the left, the number of recombinants, the recombination frequency would not assort independently from purple in the last cross that we just did. So what is the map distance between black and spec then? And basically the way we calculate this is we just add up the number of map units between black and purple and vestigial and spec. So that total should add up to 49 centimorgans. So are purple and spec linked? Are black and spec linked? And the answer to that question is no. These genes are so far apart on the same chromosome that recombination always occurs between them, and black and spec appear to be unlinked even though they are on the same chromosome. How do we know that black and spec are on the same chromosome? And the reason we know that is because we know that black and purple are linked to vestigial and vestigial is linked to spec. So that tells us that they must be on the same chromosome. So spec goes over here, 41.5 centimorgans away from vestigial. Okay, so now I'm going to have you guys do another calculation, and it's going to be along the lines of the calculation we did before using the vestigial and lobe gene, but instead of using the vestigial and lobe gene, we're going to look at a couple of other gene pairs. Specifically, we're going to look at the dumpy wings gene and the plexus wings gene. And I'm going to give you, and you're going to predict the fraction of parental and recombinant types from the cross that's carried out with these two parents. One of the parents is heterozygous for both of these alleles, and it's in the cis configuration, and the other parent is a standard test cross parent. And I'm going to give you a simplified map that you can use in calculating the offspring categories. And here's our simplified map, where 87 centimorgans separates the dumpy wing phenotype from the plexus wings phenotype. So here's three options I'm going to give you to consider for what you expect the outcome to be of this cross. And you can stop the video now if you'd like to think about this a little bit before you try and answer. But when you start the video, I'll just give you the answer. So the correct answer should be option 3. And I know in the past when I've given this question in the class, frequently the answer that people give is option number 2, where they actually think 87% of the time recombination is taking place between dumpy wings and the plexus wings phenotypes or traits. But in fact, because these genes are spaced 87 centimorgans apart on the genetic map, in fact, you're never going to see see any two gene pairs exceed 50% recombination. So because they're separated by more than 50 centimorgans, they should assort independently. Basically, you'd expect to see equal frequencies of all 4 progeny categories, just as if they were on separate chromosomes. So the correct answer is option number 3. And again, it's because they're so far apart on the same chromosome that you miss a lot of recombination events, and they just start to look like an independent assortment. All right, so I'm going to leave you with this last question. I'll give you the answers. I'll set you up here. In a certain plant species, flower fragrance is dominant over unscented, blue flower color is dominant over white, rounded leaves is dominant over pointy, and thorny stems is dominant over smooth stems. So from the following crosses, can you determine whether the fragrance gene is linked to any of the other genes? If so, at what map distance? And what I'm going to do is you can pause the video because I'm going to let you, there's 3 questions on here. You can pause the video, you can work out one, I can restart the lecture and see if your answer is correct, go on to the next one, or you can just keep the video paused and just work out all 3 of these, the answers to all 3 of these questions, and then restart the video and I'll give you the answers. So again, the first one is this one, so give some thought to this. Okay, so the correct answer for this first cross is that they're unlinked. Okay, you're seeing equal numbers of each of the 4 progeny categories, and so that's not saying us that these genes are on different chromosomes. It's just simply saying that they're either on separate chromosomes or they're so far apart on the same chromosome that you don't see any evidence of linkage for these genes. Okay, so for the second cross, again, you can pause the video if you want to work this one out, and I'll give you the answer. So for the second one, the correct answer is 10 centimorgans, and you should basically infer that from just directly from looking at the number of offspring categories. Again, you see 2 offspring categories that are much more abundant than the other 2, and the 2 that are more abundant are roughly equal in number, and the 2 that are less abundant are also roughly equal in number. Right? So right away, that's evidence of linkage. And if you calculate the number of recombinants, you find that it's about 10% recombination taking place between these genes. Okay, so now you can do the last one. And oftentimes in this problem, people will get the wrong answer. In this case, the correct answer is that you can't tell. And I'm not going to tell you why you can't tell. I just want you to look very closely at the parental, you can actually write out the chromosomes. Look at the parental genotypes and think through the cross a little bit to make sure that you understand this one. If you have trouble with this one, I'll post the answer, but I think this is something you guys can work out on your own, why it is that you cannot tell in this case. Okay, so that's it for today's lecture.